All right. First, we're gonna have that annoying bar at the top. Okay, so we're picking up with what we discussed last week. Okay, are we good? Questions? Okay. Remember what we're talking about here. We're focusing on the chemical reactions of an organism. Okay. Whether you are putting compounds together or breaking compounds apart, all of those things inside of a cell are involved in using chemical reactions, okay? For every chemical reaction, we talked about enzymes that are involved. We talked about activation energy. We talked about substrates or the starting material. We talked about the products, okay? We also, in conjunction with this activation energy or the amount of energy that has to go into that chemical reaction to allow it to proceed, there has to be what's called an energy source in those reactions. When you're working with an energy source, okay, that energy source is got to go into these reactions. And what happens, what allows us to harvest the energy from those reactions is by passing around electrons from one compound to the next, okay? So what you see here, harvesting energy requires a series of coupled reactions. And what's happening here is that one organism is donating the electron and one organism is accepting the electron. Remember, energy is stored in bonds. Bonds involve the electrons on the outside of an atom, okay? So by shifting around the electrons, that is making and breaking chemical bonds. Again, if all of this is foreign to you, you're going to have to go back into chapter 2, okay? So when you look at a chemical reaction and you have a compound that loses an electron, that compound is said to be oxidized. The compound that gains an electron is said to be reduced. And I know you're like, that doesn't make any sense to me. Why is reduced the one that gains the electron? It has to deal with the electron shells and the mass and how all of that is put together, okay? So oxidized loses the electron, reduced gains the electron. When you have a chemical reaction, when you are making and breaking bonds, it is all about shifting around electrons. What we're going to lead to today is this ultimate discussion on the electrons, and moving them around allows a cell to harvest lots of energy in the end, okay? So while it seems like these tiny little negatively charged components on each atom would be inconsequential, they're actually really important when it comes to making energy inside of a cell. Oh, but that works. Okay. So okay. Oh, we're good to go here, right? Electron carriers are these molecules that allow you to shift electrons from one component to another. Electrons are the ultimate component of allowing us to harvest energy. So shipping electrons from one component to the next is important in the process, and you have to understand what the electron carriers are. I think of an electron carrier as literally just an Uber, okay? It is like a little Uber for electrons. That is its goal. The electrons hitch a ride, they go somewhere else, and they get dropped off at this next component. That next component is going to allow you to make energy in the end. There are 3 different electron carriers that are possible in this process. Those molecules are known as NAD+, FAD+, or NADP+. Yes, they have longer names like nicotinamide adenine dinucleotide. I'm not going to write it out, and I'm not going to ask you to write it out. You're going to see it written as NAD, FAD, or NADP, okay? In its oxidized form, it's got this extra positive charge to it. So when you see that NAD plus, you know that's in the oxidized form, okay? It's waiting for the electron. When it gains the electrons, then it becomes in the reduced form. And now it goes from NAD plus to NADH, okay? H stands for hydrogen. Hydrogen is atomic number 1. It has 1 proton, it has 1 electron. So what we're doing here is we're gaining, we've got 2 electrons that are coming on here. We've got an extra positive charge to start with. So this allows all of these components to come together in this reduced form. Now it becomes NADH plus, and then it's sitting here with an extra H plus is just a proton, okay? NADH is in your reduced form. Anytime you see the H on the end, H stands for hydrogen, 1 proton, 1 electron, because it's added on to the end of this, that is the reduced form. That is the, I'm in the Uber and I got the electrons form to it, okay? So you can see NAD plus becomes NADH. FAD plus becomes FADH2. NADP becomes NADPH. When you see the H on the end, it means it's carrying those electrons. Carrying those electrons means it has to be in the reduced form. We're going to talk about NADH and FADH2 in response to aerobic and anaerobic respiration. NADPH is functioning more for biosynthesis. The only way we use this is in photosynthesis. So I always remember that when I see the P on that molecule, I know I got to be talking about photosynthesis for that one, okay? NADH, FADH2 are used to drive a proton motive force and make ATP. Those are going to be used for electron transport. Those are used in aerobic and anaerobic respiration. NADPH is going to be the one that we're going to focus on when we talk about photosynthesis, okay? When it is reduced, that reduced form represents what's called reducing power. Reducing power simply means the ability to make ATP. These guys are simply just a little Uber for electrons. They are taking the electrons to the electron transport chain where they are going to be used to make more ATP, okay? So when we put these chemical reactions together and we start talking about these bigger processes, you need to remember that yes, these molecules are coming off, but they represent important components with these cellular processes because it's going to allow us to make energy in the end, make ATP. What are our goals? Yes, it's a high flooding. Okay. Our goal is to say, okay, we're going to start with a molecule of glucose. Glucose, remember, C6H12O6. And do the same with the markers again. Okay. All of the bonds holding those atoms together are holding on to energy. The goal is to take that glucose molecule and break it all apart to harvest energy from it. We're going to talk about the processes in which this can happen. So an organism can go through glycolysis. It can do pyruvate oxidation. It can go into the TCA cycle, which is also called the Krebs cycle. It can go down an electron transport chain. Some organisms do glycolysis and then switch to fermentation. The goal is to say, how can I break up this one molecule of glucose at the top? Some of these processes directly produce ATP. So you can see there's ATP coming off of glycolysis. You can see ATP coming off down here from the TCA cycle. But what's actually more important is all of this reducing power that you see in these little boxes here, okay? That reducing power allows you to make more ATP later. Do you see how this reducing power is funneling in here to this respiration process? This one coming off of here is funneling into respiration. Some of that from up there can funnel down here. This allows us to make lots of ATP in the end, okay? Remember what we said, when ATP is a direct product of a chemical reaction, it's called substrate level phosphorylation. Phosphorylation means what again? Adding on a what? Adding on a phosphate group. So when we add on a phosphate group, we go from ADP to ATP. You can see here, what comes off of glycolysis is labeled substrate level phosphorylation. What comes off after the TCA cycle is labeled substrate level phosphorylation. The ATP that's made after respiration, that simple electron transport chain, is all coming out through this oxidative phosphorylation process. These are all catabolic processes. We're starting with glucose and we're breaking it down. The goal of breaking it down is to provide energy, make some direct ATP. To make this reducing power goes to make more ATP. We also make extra precursor metabolites and intermediates. You see some of those up at the top. You can see some of those down here. A precursor is simply, or an intermediate, remember, is a compound in the middle that can be used for something else. We haven't gotten rid of it. We've produced it as a byproduct. And what else can we do with it in this particular situation? We don't give off any waste products. We don't throw away anything unless we absolutely don't need it. It says, what else can we do with it inside the cell? Okay. Are we doing okay? We look like we're not having fun for a Tuesday. It's a lot of like staring. If they told me this wouldn't happen. Yes.
What is this under? Like which is this under unit 5?
It's unit 4.
Okay, so
Yep. Right. Trying this again. Okay. And we'll leave our controls up there because maybe that. Okay. This table summarizes what we are going to spend 20 minutes going through. Okay. This is a table from your book, all on this unit, but it breaks apart each process: aerobic respiration, anaerobic respiration, and fermentation. What they are using and how much ATP is produced. Okay. So we're going to first talk about respiration. Respiration comes in 2 forms: aerobic, which means with oxygen, and anaerobic, which means without oxygen. What that term really means comes back to the final electron acceptor in the process. Aerobic respiration, oxygen, molecular oxygen is the final electron acceptor. In anaerobic respiration, it's going on to any other molecule that is not molecular oxygen. So when we say molecular oxygen, it's in the form of O2. That is all coming back to this periodic table of elements and the number of electrons in that molecule. Because of oxygen being atomic number 8, it is not stable unless it has 2 oxygen atoms next to one another. That makes a stable atom, okay, with the electron shells. Aerobic respiration, molecular oxygen or O2 is the final electron acceptor. Anaerobic respiration, it's going to go on to any other molecule. Nitrate is common, nitrite is common, sulfate, but that's going to be entirely dependent upon the exact organism that uses it. So do not memorize any more details other than it's going to be on something other than molecular oxygen, okay? Because every species will be different. And when they run out of some source, they can switch to something else. So it will be different for each organism for where it's at and what it's doing at that particular point in time if they're using anaerobic respiration, okay? Just know it's using something else. Why are we saying terminal electron acceptor? This is going back to that electron and those Ubers, right? Who's getting the electron in the end? Think of electrons like a little hot potato. You pass them around, you pass them around, you pass them around. That process of passing them allows those components to make energy. But in the end, someone has to be stuck with the hot potato, okay? You don't just, you've played this game maybe, okay? You don't just, or musical chairs. You don't just, oh no, that's a bad one. Hot potato. You don't just like toss the potato on the floor when you're done. Someone gets stuck with it. In all of these processes, we don't just kick the electron out and just leave it somewhere. It's got to be left with somebody, okay? So it's got to go somewhere. In anaerobic respiration, oxygen is the hot potato that gets, is the one that gets left with that hot potato that is the electron. In anaerobic respiration, it's going somewhere else, okay? So far, so good, okay? What happens to create these processes, or what happens to create our energy? When we have aerobic and anaerobic respiration, those two are large processes that do both substrate level phosphorylation and oxidative phosphorylation to make ATP. Substrate phosphorylation is the direct production of ATP in some process. So you can see we're going to make 2 ATP from glycolysis. We're going to make 2 ATP from the TCA cycle. Our total is going to be 4. Anaerobic respiration, the number is going to vary. I'm never, ever, ever going to ask how much ATP is made in this process of anaerobic respiration, because it's going to be different for every organism. It's going to be different for what they're using as that terminal electron acceptor, okay? So if we're looking at any numbers here, we're focused on aerobic respiration and we're focused on fermentation. Once we've made all the ATP we can from substrate phosphorylation, all of the reducing power goes over to the electron transport chain. And that electron transport chain allows you to make more ATP using electron transport, which is also called oxidative phosphorylation. Look at this. In aerobic respiration, you only make 4 ATP through that direct production, but you make 34 ATP in electron transport for a total of 38. Okay? So electron transport is super important to any of these cells because that's the way you're going to make the most ATP inside the cell, okay? Do you see how over here it says uses an electron transport chain? Electron transport chain means it's completing oxidative phosphorylation, okay? So this column and this column over here are going together. Yeah. Yeah. So substrate level is the direct production of ATP when it's coming off of a chemical reaction. So if any chemical reaction directly gives off ATP or biochemical process gives off ATP, that's going to be substrate level phosphorylation. If the only way you make the ATP is through an electron transport chain and tossing around electrons, then that's going to be oxidative. Aerobic and anaerobic respiration use both processes. Yeah.
Can you say the substrate level one more time?
So substrate level phosphorylation is direct production of ATP in a chemical reaction. Yes. Doing okay. We're gonna get there. We're gonna keep repeating it and we're gonna go through it. Okay. I'm like stairs.
The phosphorylation is creating ATP regardless of type.
Yes. Regardless of type,
What phosphorylation means.
adding so it's phosphorylation itself means adding on a phosphate group. So going from ADP, adding on a phosphate gives you ATP. So you can directly do that, substrate phosphorylation. So directly adding on the phosphate group in a chemical reaction to make ATP. Or you use a electron transport chain to help you add the phosphate group and make the ATP. We're gonna keep going through it, promise. We're also going to talk a little bit about fermentation. Fermentation is an important process that prokaryotes can use to make some ATP. But fermentation is a completely different process because it's only going to use glycolysis to make substrate phosphorylation. It's only going to give you 2 ATP total. You're not doing electron transport, which is why over here it says no, okay? And you only make 2 ATP in the end. So if a cell has a preference and it wants to make the most ATP, what should it use? Aerobic respiration, anaerobic respiration, or fermentation? Aerobic, okay? Aerobic makes the most ATP for any living cell. If it has a choice, that is the preferred way to go. Okay.
That's like pretty much like for any type of cell.
Exactly. Our cells do aerobic respiration. That is our preferred method of making ATP for our cells. Okay. When our cells run out of oxygen, they switch to a different form that is less productive. Okay, yeah.
Fermentation uses
Glycolysis only. Yep. All we're doing here is talking about catabolic reactions. What do those reactions do again? Break things down or put things together? Break down. Okay, so we're talking about the breakdown of compounds. Breaking down those compounds, most commonly the compound is going to be glucose. Okay. And our goal is to say, okay, how can I break this down to get the most out of it? I want to make intermediates to help me make ATP. Those intermediates also generate reducing power. But remember, this just means the ability to make more ATP later. Okay. Intermediates and end products all become precursor compounds that can be used in other places. I break this apart. What do I do with it? Okay. You go to I went to this place called Level One over the weekend. Okay. I'm feeding money into the change machine, because let's get quarters so my kids can all play pinball, right? Whatever quarters they don't use, I'm like, oh, I got, I'm gonna buy a beer. Here we go. Like, this is for me, this is for them. All the money got used, but it was like, what do we do with it, right? Catabolic processes say we have this one starting material. We're gonna break it up into smaller things, and then each of those components gets used. We don't toss anything away that we don't, that we can't use for something else later, okay? So the goal is to say, I'm gonna start with glucose, but I'm gonna make as much energy out of that as I can. I'm gonna squeeze every last ounce of that energy out of this molecule. And then I'm gonna take everything left and say, what else can I put together to use this? Okay. Here is another way of looking at this process. What is on the table and what is in this diagram are essentially the same thing. But sometimes the visual is a little bit better here, okay? So look at this. Do you see how these two line up? They are almost exactly identical until you get to the end. And oxygen is the final acceptor with aerobic respiration, and something other than oxygen is the final electron acceptor with anaerobic respiration.
And this is still for substrate level.
So substrate level is going to be when we directly see it come off. So here, this ATP is substrate level. The ATP up there is substrate level. When we're using that electron transport, that's oxidative phosphorylation. So the total down here is a combination of the two.
Okay.
Okay. Yep.
Would you say the substrative is like just the glycolysis part?
Glycolysis and what's coming off of the Krebs cycle or the TCA cycle. Yeah,
Can you explain a little bit about Erickson's split chain?
we're gonna go through it. We're walking through the whole thing in a little bit. We're just talking general now. Okay. $38 ATP in aerobic respiration. Anaerobic respiration is less efficient, somewhere between 2 and 36. It's going to vary for every organism. Okay. When it comes to fermentation, fermentation is a little bit different. It starts out the same. Do you see how we still begin with glucose? We still do glycolysis. We're ending with pyruvate, and then we go under a fermentation pathway. Organisms that cannot complete this other stuff are organisms that then are forced to do fermentation. They don't have the ability to go into the Krebs cycle. They don't have an electron transport chain. When all of this is missing, that's when they go here instead. It's okay, but it only allows them to make a small amount of ATP because glycolysis is the only place we're making that. Yeah.
For catabolic reactions, are we only going to be talking about the breaking down of glucose? Because I know there's other OSAs, but.
Right, so we're gonna hit a little bit on that later. Because some of those other sugars can be broken down to then make glucose to then head into this process. Glucose is the easiest sugar to break down. When you don't have glucose, you break something else down instead to go into part of this. And we're gonna get to that. Yeah. It's a form that's been converted. So that's more of a storage molecule. So it's not completely a glucose molecule.
Fermentation only produces 2.
Yes.
Okay.
We're going to focus on 4 main components that allows us to break down glucose and to make energy. We're going to take this step by step, okay? The first one is glycolysis. This is where we begin the breakdown of glucose, okay? Breakdown is also referred to as oxidation. Remember, oxidized and reduced. What do those forms mean, right? Reduced means we're gaining electrons. Oxidized means we're losing electrons, okay? So when we say oxidation, we're losing electrons because we're breaking the molecule apart. So the first part of this process of both aerobic and anaerobic respiration is going to be glycolysis. It starts the breakdown of the glucose molecule. After that biochemical process is finished, then we go into what's called the transition step. The transition step is also called pyruvate oxidation, okay? After we've completed pyruvate oxidation, we're going to go into the TCA cycle. This is also referred to as the Krebs cycle. And then lastly, we go into the electron transport chain. What we are talking about here are these processes in prokaryotes. You should have at some point already went through them for a eukaryote. Okay, so when you went through this previously in your other biology classes, it was learning the processes in a eukaryotic organism. Okay,
You said the TCA cycle referred to as one.
Krebs. So you got two different names for half of these processes, but they're the same process. Okay,
An electron transport chain.
it's just electron transport. Or ETC, sometimes you'll see it. ETS, occasionally electron transport system, but electron transport.
Is there a different name for glycolysis?
Nope. I know. That one they leave alone, okay? You are not memorizing every chemical reaction, okay? The goal here is to show you the chemical reactions so that we can talk through. Run out of board. Okay. So this is a series of 10 chemical reactions, okay? A few things to point out. Do you see how we are starting with glucose? The glucose molecule is depicted showing you the carbon atoms. So you've got 6 carbons across the top. That's where we're standing for glucose. We're following the carbons in this process. Do you see how at first ATP goes to ADP? Remember, making ATP is creating that energy. In this case, we're breaking it down. So there has to be energy input to start with in order to begin this process, okay? This is like one of those, if you run out of money, it's hard to then go back to making money. Like money has to go in a little bit to bring money out. Similar to what's happening here. You can't begin glycolysis without energy input. If a cell runs out of energy, it has 0 ATP left, it has nothing available to begin this process, okay? The phosphate that comes off of here, do you see how it's now attached to this molecule? We go from glucose to glucose 6-phosphate. So we put energy into this chemical reaction. We go from ATP to ADP, but our phosphate just comes onto this molecule here. Guess what? It happens again, okay? We do another process where we break down ATP to ADP, and now we're adding on another phosphate. Now it actually becomes fructose 1,6-bisphosphate. Don't memorize the molecules, it's not important, okay? After this molecule actually gets broken apart into 2. This becomes 2 molecules that are exactly the same. And from this point forward, the processes happen identically on either side of this pathway, Okay. So 2 ATP have to come in. Look at what comes out, okay? Do you see this process here? We're going from ADP to ATP. So we're making ATP over here. Guess what? We're making it over here. We're doing the same thing down here. We're making ATP here, but we're making ATP here. Now, 2 slides ago, I told you the amount of glucose that's made in this reaction is 2. And it said a net of 2. We make 4. But because 2 of them have to come into this reaction, our net gain is only 2 ATP in the long run, okay? We also make this molecule of NADH. Remember, when it's got the H on the end, it is reduced because we've gained those electrons. When it's in this form of NADH, we have our little Ubers that are taking our electrons to other parts of this chemical process. We are finishing with 2 molecules of pyruvate in the end. Okay, yes, sir. 2 are gonna 2 total. 1 on either side. Okay. All fine, okay? Within these processes, you want to know, what is my starting material? What are my products? You also want to understand the goal of the process, okay? Okay, so if we're gonna do glycolysis, and it makes it worse that I'm left-handed, okay? So our goal is to begin the breakdown of glucose. We're taking this molecule, we have to start breaking it apart, okay? Our starting material is what? We need 1 glucose. What also has to come into this process? 2 ATP, right? So then our products are the 2 pyruvate. How much NADH? 2. And then we have ATP, okay? You will see this written different ways. We're producing the 4, but remember, we only come ahead the 2, okay? So if you're gonna write our starting material as 2 ATP, our product is gonna be 4 ATP. You will also see this where you don't show the ATP coming in, and you know that our net is just 2 ATP, okay? So I think it's easier to just know that it's 2, 2, and 2. Because that's what we're gonna, ultimately, when you look at the product of this process, 2 of those get us to a neutral amount of ATP, and then 2 become the benefit, okay? So we're got a net total of 2 ATP that's coming off. I don't care that you know any of this. I need you to know this, okay? Glucose is that 6-carbon sugar. What we've done in the end is turn it into 2 3-carbon molecules. We still have 6 carbon atoms. You see 6 blue circles up here. We still have 6 blue circles down here. The carbon hasn't went anywhere, okay? All we've done is break this molecule up into two components. Now, each one of these components is moving into our next phase. Okay? So what we're doing here is to take that glucose molecule we started with and start our breakdown. We went from 1 6-carbon molecule to 2 3-carbon molecules. That process we did in 10 chemical reactions, but it allows us to make a little bit of NADH, which is that reducing power, and it allows us to make 2 net ATP that we can use to do whatever else we need to inside the cell. So far, so good? I wish we could just be consistent with our colors, okay? Just imagine that these are little blue circles, okay? Pyruvate and pyruvic acid are the same thing, okay? The next one is called the transition step. The transition step is also called pyruvate oxidation, okay? The goal of this is that the product from glycolysis, pyruvate, can't just go into the TCA cycle. It's not compatible. It's got to be converted into something else. So this step links glycolysis and the TCA cycle together. We go from a 3-carbon molecule that is pyruvate or pyruvic acid, and we convert it into a 2-carbon molecule that is known as acetyl-CoA. We get no ATP out of this process. This is not energy generating. But what it's doing is furthering along our precursor metabolites to create this molecule that goes into the next step. So we make a little bit of NADH. Remember that NADH is that reducing power. Because we have lost a carbon, the carbon is coming off in the form of carbon dioxide, CO2. While the process is different, it's almost easy to think I breathe in oxygen, I breathe out carbon dioxide, right? You bring it in, but you bring it out in a different form, okay? Again, the mechanics of how that works are different. But I need you to know that every time we break off a carbon, carbon dioxide is released. And I know, it's coming back to your chemistry. Carbon is atomic number 6. Means it's got 2 electrons in the first shell. It's got 4 electrons in the next shell. It's not a stable atom. Everything has to be in a stable format. Carbon dioxide is the most stable format. I can almost guarantee you between a homework assignment or an exam, you will be asked. Every time a carbon atom leaves, what is its form? And your answer is going to be CO2, carbon dioxide. Every time carbon comes off, it's not just going to be an individual carbon atom floating around. It's going to combine with other atoms to make a stable molecule. One last component. This is a chemical reaction where 1 pyruvic acid comes in and 1 acetyl-CoA comes out. How many pyruvic acids did we create in the last process? 2. So this has to happen twice. The first pyruvic acid comes in and 1 acetyl-CoA comes out. The second pyruvic acid comes in and the second acetyl-CoA comes out. When you look at a chemical reaction, when you're shown a figure of a chemical reaction, it's always going to show you the figure, based on what your starting material is, okay? In this case, we got to remember our starting material is the 2 pyruvic acid.
The transition step happens twice.
Yes.
Okay, because there's two pyruvate.
Yes. So you want to remember, so yes. She's like, I think this is a homework question. Is it in the homework or the notes?
Notes.
Oh, it's in the homework too. Exactly. So the transition step or pyruvate oxidation happens twice for every glucose you start with. If the question said, how many times does the transition step happen for each pyruvate? Only once. So that's where it gets tricky. You'll have to remember what Smith's starting material like. Yep. Yes. I can't possibly do this without it. It's too short. Okay. All right. So transition step. AKA pyruvate oxidation. Our goal. Oxidized pyruvate, okay? Oxidize is just another word for breakdown, okay? So here we said begin breakdown of glucose, you could say breakdown pyruvate, okay? Breakdown and oxidation are the same thing, okay?
Can you send your oxides and glucose for glycolysis?
So because the glucose is gone, you don't have any of that left. You could say continue the oxidize of that molecule. It originally started off as glucose, as long as you understand that that's the part of the molecule that's continuing. Yep. So your starting material, remember these are pathways. So it's one part of the product over here is coming to be the starting material over here. And what is that molecule? The 2-pyruvate, okay? We're going to take those 2-pyruvate and we're going to convert them directly into acetyl-CoA. 2-acetyl-CoA. Two NADH.
Two CO2.
And 2 CO2, okay? Think about constantly making sure we've got all the carbons accounted for. And then, you know, wait a minute, I know there's only 4 total up here. So I know the other 2 have to be down here, okay? So far, so good? I told you we will get through this, okay? Starting to look a little better. There were a lot of worried faces here for a while, okay? I know, you're still worried. Oh, won't it come back? This projector gives me more grief. The TCA cycle, tricarboxylic acid cycle, is also called the Krebs cycle, okay? This one is a little different because it's a cyclical pathway, right? Your book and I are not aligned when I see these diagrams, okay? You have the transition step. This diagram from your book is showing you that transition step at the top so that you understand the acetyl-CoA from the one before is what's headed into this particular pathway, okay? Again, you're not memorizing the chemical reactions. The goal is actually to say, okay, I got these 2 carbon atoms left. I need to break these apart to get as much energy as I can out of this molecule. This is the last part where we're finishing that breakdown, okay? This is completing the oxidation of the glucose molecule, to Rachel's point, okay? It's also known as just oxidizing the acetyl-CoA, okay? All we're doing is breaking it down. So the acetyl-CoA joins with our 4-carbon oxaloacetate to make a 6-carbon molecule. The chemical reactions that follow are literally just to break apart those atoms and rearrange the molecule. In this process, look at how fun this is. We're gonna make 1 NADH, 2 NADH, 3 NADH. That's a good chunk of reducing power. Do you see how we give off carbon dioxide here and carbon dioxide here? There were only 2 atoms of carbon left. So by the time we're halfway through this process, that carbon is gone. It's out in the form of CO2. It's out of here. But the beauty of this cyclical process is that in doing those reactions to regenerate our starting material, we actually make ATP. And we're going to end up making FADH2 in this case, more reducing power, and that last NADH, okay? We're directly producing ATP in this process. So is this one substrate phosphorylation or oxidative phosphorylation? Substrate. Awesome. Okay. And how many acetyl-CoA do we have? 2. So how many times do we do this? Twice. Yeah. So when we directly make ATP off the process, that's the substrate. So when you see that ATP come off of a specific chemical reaction, that's what makes it substrate phosphorylation. Mhm.
Or like the difference is like whether it has like oxygen or
So aerobic versus anaerobic is coming back to the final electron acceptor at the very end. If it's aerobic respiration, the electrons are going on to oxygen. If it's anaerobic respiration, they're going on to something else. Exactly. So if you learn it for one, you know it for the other. Doing okay? You know where I'm going with this, okay? We're going to divide this up. Then we're going to draw out electron transport. Yeah. Okay. So.
It's like chemistry over.
Is.
Yeah, chemistry was awful.
That was awful. All right. Krebs, TCA, same thing, okay? Our goal here is to complete the oxidation. Acetyl-CoA. You will also see complete oxidation of glucose, okay? I'll write it over there. Okay. Our starting material. Is what? 2 acetyl-CoA. What are our products? 2 ATP.
3, 6.
6 NADH. 2 FADH2. and 4 CO2. We started with glucose, that 6 carbon molecule. Do you see how between these 2 steps now we've got all 6 carbons accounted for? Yeah. Yeah. So we already doubled them. So if it was to say what happens in 1 round of the TCA cycle, you'd have 1 acetyl-CoA going in, 1 ATP, 3 NADH, 1 FADH2. We're drawing this out for 1 glucose molecule. So for that 1 glucose molecule that starts, these are all your products.
And then as you're adding glucose molecules.
Exactly. So another glucose molecule goes in and keeps going, and it just keeps all going. Yeah. But we already put them in here. So the transition step, each 1 pyruvate goes in. So total for the glucose, you're gonna have 2 pyruvates, which gives you 2 acetyl-CoA, 2 NADH, and 2 CO2. So we already doubled it here, and we already doubled it here. This is the total that's made for the 1 glucose molecule that we started with.
Can you explain how we get the NADH? Like, I understand the CO2, I understand,
Yes.
I don't understand how we get the NADH.
So the NADH is coming off, it's a redox reaction. So an oxidation reduction reaction that's just transferring electrons.
Okay.
So that NADH is just taking electrons from that rearrangement and then putting them into a carrier molecule. So the NADH is just the Uber that's leaving off the process with what's been made. Hold on, there's a diagram.
Okay, so even though we have all of these products, for each one, we're only using 1 to begin the next step.
For only using 1 to begin the next one. So
And then the other products are used.
exactly. So the CO2, if the CO2, if it can't be used by the cell, it'll get converted or it'll get released as a waste product. The ATP, it's going to directly use that energy to make molecules in the end. We're going to talk where this NADH and FADH2 are going in just a second. Anytime you have extra components, they then get funneled into other spots. Or let's say we do it with 1 glucose and we've made enough ATP and the cell goes, okay, listen, I don't need any more of that. So the cell then takes those molecules and converts it into something else. Or maybe we've made this pyruvate here and it goes, I don't need to do all of this because I have too much. It can take this pyruvate and shuttle it into something else instead.
But so everything that we're not using in the next step is compiling
Is
for whatever
compiling for
else or waste. So like we have 6, 2, and 2, so we have 10.
Yes.
Okay.
Yes. Whatever doesn't get used in the next process, then either has to go somewhere or get used by something else inside the cell. The ATP will get used to do anabolic reactions. And then we're going to talk about all this reducing power in just a second. When we say respiration, the respiration is coming to the last step, and that is in the electron transport chain. Where we're going with this is to make lots of ATP. Remember that diagram at the beginning? 2 ATP by 4 ATP total by substrate level phosphorylation, 34 ATP by oxidative phosphorylation. We said the electron transport's the biggest deal with that. That's where all of this is headed, okay? Oxidative phosphorylation is going to use all of those extra electrons that we put in the little Ubers, NADH and FADH2. And it's going to use those to generate a proton motive force and then make ATP in the end, okay? It is just a game of hot potato of passing energy, electrons, down a gradient to allow us to make ATP in the end, okay? the electron transport chain, where all of those other ones that we just talked about were these these chemical pathways, right? What we're talking about now is literally that game of hot potato of continuing to pass the electrons. By passing around the electrons, that allows a series of processes to occur, which allows us to generate ATP in the end, okay? This is called electron transport. But when you transfer electrons, you also have to transfer protons. The two of those components go hand in hand. So what we're going to talk about here is creating a proton motive force. There's always kind of a yin and yang balance, right? We talked about staining and we said opposites attract. It was all about the negative and the positive. The same thing happening here. Those negatively charged electrons don't just flow by themselves. They're actually going to move with positively charged protons along with it. Remember what we said, NADH. The H on the end stands for hydrogen. Hydrogen is one proton, one electron. So not only are the Ubers carrying the protons, the electrons, they're carrying the protons as well, okay? So what's going to happen? This happens at the cell membrane of a prokaryote. When you learned about this in eukaryotes, you learned about mitochondria and how it all happens around that pre-stay inside. Remember, prokaryotes do not have mitochondria. So glycolysis, TCA cycle, transition step, they all happen in the cytoplasm. They happen in the cytoplasm of a prokaryote, and then those electron carriers approach the membrane. Do you see how NADH is coming up to the membrane here? Okay. The NADH comes up to the membrane and it leaves in the form of NAD. So it's kicking out not only the electrons, but protons as well, okay? Both of those have a path. The electron is going to go through the middle of the membrane, okay? Do you see how it's joining here with oxygen? It joins with oxygen, it ultimately ends up creating water in aerobic respiration. So what you're looking at here is aerobic because it's oxygen that's depicted here, okay? So the electron flows through the middle of the membrane and hits these carrier molecules. That electron going through allows the protons to go from the inside to the outside. Do you see how this says H+? H+ is just a proton. A positively charged hydrogen ion means it has one proton and 0 electrons, okay? So the protons go out. The protons go out. It creates what's called a proton motive force. All a proton motive force is a gradient where there's large amounts of protons out here and fewer amounts of protons here. If this is the cell membrane and this is the inside of the wall, what's way up here? What structure? It's on the outside of the cell membrane. Cell wall, okay? So the protons are going to build up here and the cell wall is up here, blocking them in. What do you remember about your concentration gradients? Things want to flow from what to what? High to low, high concentration to low concentration. All a proton motive force is a high concentration out here. And those protons go, whoa, I need to flow to a lower concentration. The protons are going to flow back through this particular carrier in the membrane. Do you see how this ends in ASE? ATP synthase. What kind of molecule did we say that has to be if it ends in ASE? It's got to be an enzyme, and enzymes are made of proteins, okay? So the protons flow back through this particular enzyme, and that particular enzyme allows for ATP to be produced. The only way this ATP is made is if electrons flow, protons build up, and then the protons come back in. This is oxidative phosphorylation.
What did the NADH and the FADH2 do?
Awesome question. Because this goes back to being NAD, it goes back over to glycolysis. And it'll just shuttle another proton and electron and bring it right back here. So it's just like an Uber that goes right back to where it was at before and brings more back into the party.
So the proton has to go back through the cell membrane to complete the cycle.
Exactly. The proton, the flow of the proton is actually what allows ATP to be made. I know it sucks. It's called the electron transport chain. The electron transport chain does nothing without the protons along with it.
Is there a specific path that the protons travel that we need to know?
No. Because that can be different for every organism with different carrier molecules that are in the membrane. That enzyme ATP synthase kind of sounds like synthesis, right? Okay. That is an important enzyme because that's what's going to allow us to make ATP in the end. Believe it or not, for one NADH molecule, we're actually going to pump out 10 protons. Okay. For every one NADH, we're going to make 3 molecules of ATP. For every FADH2, we're going to pump out 6 protons, and we're actually going to make 2 molecules of ATP. So look at this. We have 2 molecules of NADH over here. Each one of those is going to make 3. So from these 2, we're going to make 6 more ATP. So that's why a small amount here translates into a big amount of ATP once you're finished with electron transport. Okay. So like 10 slides ago, when we said you make 38 ATP, where is it coming from? You make 4 ATP total in that substrate phosphorylation. 2 from glycolysis, 2 from the TCA cycle, under here somewhere, okay? Then everything shifts to the electron transport chain. And you're gonna make 34 total. 6 are coming from the 2 NADH that are in glycolysis. 6 are coming from the 2 NADH that are from the transition step. 22 are coming from the NADH and the FADH2 that are from the TCA cycle. So that's how we end up with 34 total from oxidative phosphorylation. The total yield of ATP in aerobic respiration in a prokaryote is 38, is the total maximum. When you look at diagrams, processes, a regular textbook showing you eukaryotic respiration, it's going to tell you that number is 36. The number is 36 in a eukaryote because they actually lose 2 more ATP, pushing it across the mitochondria, okay? We are focusing on a prokaryote. Our maximum number is 38. All right. Let's do this. So where is it all going? Can you all see this? I draw it a little bit taller. Okay, this is the cell membrane. All of this, and these are all headed here. So our goal is to bring it all right up here, okay? There's a carrier in the membrane. Carriers in the membrane, okay? This goes from NADH back over to NAD plus. Gonna draw it generally, we're giving off electrons and we're giving off protons, okay? The electron goes through the middle of the membrane. It's gonna come back out. Terminal electron acceptor is here. Your protons are going up and out. What happens is we're gonna pump more protons out. We're building up protons, 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 okay? The protons are gonna come back over here and they're gonna come back in here. This is our ATP synthase. This allows us to go from ADP to AT. To the tune of 34 of those, if it's aerobic. We'll get out of the way. Yeah.
So like for every one NADH, how much ATP?
So for every one NADH, we've got 3 ATP coming back this way.
Is it the same for
FADH2 is double,
one of the 6?
right? Well, 6 protons pumped out per FADH2, that equates to 2 molecules of ATP. So it's not like a 1-to-1 transition, which is why, so all of this is coming up here. This will go from FADH2 over to FAD. And we're giving off electrons from here and we're giving off protons from here. All of that is coming this way to give us that total of 34. Yeah. The product that ends up, in this case, it's gonna, and if it's aerobic respiration, it'll be oxygen. So it technically goes into 1/2 of O2, which then gets converted into water. So the final product there will end up being water.
Is aerobic another name for electron transport chain?
Say that again?
Is aerobic another name for electron transport?
So
chain?
respiration is another name for electron transport chain. When you see respiration on the end, that tells you we have to go through oxidative phosphorylation, and that means we have to be doing electron transport. Yeah,
Would we expect to like see this on the exam?
yes.
Like fully explaining it,
Fully,
knowing the
yeah.
numbers, like that. How do you mean by that?
So you can be asked, what's the starting material of glycolysis, or what are the end products of glycolysis, or for each. We're gonna have a cell doing aerobic respiration. It's gonna complete glycolysis. What are the goals, products, starting materials? And then it may say, explain the electron transport chain, being sure to point out where protons and electrons are coming and going and how much ATP is making it. I won't make you account for every proton and electron and how, but if you gotta give me that totally. How are we feeling about it? Better? Aerobic and anaerobic respiration go through the exact same stuff. However, anaerobic respiration uses different molecules in the end, so therefore it is less efficient, okay? I don't care that you memorize any other terminal electron acceptors at this point for this stuff. It is simply that when you see it goes through anaerobic respiration, you know it's not making that maximum amount of ATP, okay? These molecules give off other compounds, they do other things. So for example, E. coli growing in your gut, it actually uses a slightly different carrier molecule in the membrane. So these molecules that we drew embedded in the membrane there, different molecules, it's got a different carrier component. It gives off vitamin K as a product from that process. You absorb the vitamin K and you use it. So every product that we've made has got to either be used by the cell or it gets given off as some sort of metabolite that the organism can secrete. In nature, other organisms can use those products. When it happens inside of your body, those products can function to your benefit. They can also function to your detriment. Yeah.
So when you say that the basically like the process is the same except for the end, you're meaning like the electron transport chain, right?
Right, the final electron transport terminal electron acceptor differs a little bit. You're still dumping out protons and electrons. So if you've got this process, you still have the main component down.
It's
What
just that-
that molecule is in the membrane might be different, but I don't care that you know that anyway. We doing okay? When you see the word respiration, it means the organisms go all the way through to the electron transport chain. Fermentation, say respiration anywhere in it. No, it is a different process entirely. In fermentation, you do glycolysis, and then you take that pyruvate and you convert it into one of two other components. Fermentation is an anaerobic process, but it's not a form of respiration. There are two different fermentation pathways. There is a fermentation pathway that can make alcohol. In this case, you're showing it with ethanol. It can also make acidic products, in this case known as like lactate, okay? Each cell has the ability to choose, what do I want to do with it? Do I go down this process of making acid or do I go down this process of making alcohol? That will be different for every species. In general, most bacteria, okay, are going to go down an acidic pathway. Some bacteria go down an alcohol pathway.
But it's always acidic or alcohol.
It's always acidic or alcohol. Those are the only two options. Here's another way of looking at it that might make it a little bit easier, okay? We start with glycolysis. We still finish with the 2-pyruvic acid. This process is the same, just like what we talked about before. But then the pyruvate, instead of going into the next step here, they then go into fermentation in organisms that do this, okay? Mammalian muscle can also go under a fermentation process when we run out of oxygen. So for example, like marathon runners, you run out of oxygen in your muscle, it switches to a fermentation process and produces acid in there. Runners get cramps in their legs and their muscles because it ends up converting, they don't do respiration in that situation when they've run out of oxygen in the muscle. Okay? Why am I just saying, understand it can form acid or it can form alcohol? And that is because species can do different things, okay? Yeast tend to make alcohol as a byproduct, but Clostridium species also tend to make alcohol, or you have these other ones that tend to make more acidic products on the other hand, okay? It will be different for every species. So don't memorize pathway, don't memorize chemical reaction. Understand that if you're gonna do fermentation, those are your two possible products, is something that's making acid or something that's got alcohol in it, okay? We've spent a lot of time going through this. I want you to understand glucose is the easiest sugar to break down. Glucose is a simple sugar, and almost everything that you take in and these bacterial cells take in is all full of glucose. If glucose isn't the primary sugar available, those organisms can break down other sugars to form glucose to then enter into this pathway. If the organism runs out of sugars entirely, it can break down something else. A common pathway to then do this is to break down lipids and fats. You break down lipids and fats, they can make acetyl-CoA, they can then go into the Krebs cycle. You can break down proteins into amino acids to then produce pyruvate. The only way you make 38 total ATP is if you go through that entire process from start to finish. If you run out of glucose, and let's say you break down fat and you make acetyl-CoA, all you're completing from this point is the Krebs cycle. So you get the little bit of ATP from the Krebs cycle, but you get the NADH and the FADH2, and that feeds over here. So you're not making 38 ATP, but you're still making some, okay? If you break down amino acids instead and you make pyruvate, cool, glycolysis is out. But everything downstream of glycolysis is going to give you ATP, okay? So this is a way of giving a cell flexibility. Glucose is the easiest thing to break down and it completes the process. If it doesn't have glucose, it doesn't just roll over and die. It says, what else can I use to make ATP? I have to have a constant supply of ATP. What else can I use?
So is
You
this,
run out of money in your bank account, you start selling off your things, right?
so is this like a preferential order, like glucose, more complex sugars, fatty acids?
Typically, yes. Part of that will also depend on what the cell has available, but in general, that's the way it's gonna go. Yeah. Correct. You got it. All of your cells are also doing this process. This is literally the exact same thing as the last diagram, just putting it in a different way, okay? You can break down all kinds of other components to feed into parts of this. You have to make ATP. So yes, glucose is the easiest starting material, but when you don't have glucose, you can use something else. I like this one because it's showing you on the left here how you can take polysaccharides or disaccharides and break them apart to their simple sugars. And then their simple sugars can feed into this process. All right. We're a little behind, okay? I don't know that I can just stop this. We're just not even gonna touch it. We're just gonna let this go. Okay. We're gonna take a break, okay? Because I know you're all gonna hate this. We still have to get through photosynthesis. So photosynthesis is the last part of Unit 4. And then we're gonna talk through microbial diversity as we finish, okay? So It's 10:30. Come back at 10:40-ish, okay? We're gonna finish photosynthesis and talk to Berkeley before we're done for the day. I'm just gonna leave this up.
What are some other methods to go through this? Like outside of the guided notes and lecture and whatnot, like.
So there are
I think maybe
videos.
I might need this explained.
Yes, there are short videos on Blackboard, taking each process and separating it. So there's a video just showing you glycolysis. There is a video just showing pyruvate oxidation, one on the transition step, one on electron transport. You can find a lot of videos on YouTube. Most of them, however, are for a eukaryote. They're not for prokaryotes, but they'll be good for giving you the general pattern. There is some college class somewhere that makes them put this into like song. Like no joke, if you search like respiration song or like respiration 80s rap, like Baby Got Back to do going through that, I kid you not, it's hilarious. But however it works to get you to like be able to recall it. Yes, so the videos can be helpful. Just take some of those with a grain of salt, because you'll see mitochondria and you'll be like, talking about the mitochondria. It's the same process, it's just in the cytoplasm. The book is helpful, but it's dry text trying to walk you through this. So I think drawing it out like we did here or watching a video are the better ways to see.
Cause I used the book for the last unit and I felt like that was helpful, but I think it was helpful more at the end when I had more of an understanding of
It does a good job of working through it. It's just a lot. So the book, you've got the book. You could find a video. There's videos in Blackboard itself. I think it's helpful to draw it out. But that's how I learn. So,
I have a
Yeah.
question. Yeah, are we focused more on prokaryotes, just prokaryotes for these?
yeah,
units?
because it's microbiology. So prokaryotes.
Oh, yeah, okay.
Yeah.
it. You didn't say it like.
Yeah. Thank you.
Yeah. The immune sisters are pretty good. Or the amoeba sisters? The amoeba sisters. Their immune system stuff, when we get to that, is a nice one.
Are you
Amoeba.
in bio?
Yeah, they have like their own little YouTube channel. They're pretty fun. Some of their videos are a little long, but they're pretty good. What's that?
What's the YouTube's name?
It's the Amoeba Sisters. Amoeba, AMOEBA. They do all kinds of drawings. They have little like cartoons that you can like just static cartoons. Yes, we're working on it. Sorry. Yes, we're working on it. All right, thank you. Yep. Oh, well.
The final exam's
Yep.
comprehensive, right?
Correct.
Will we ever be able to see what we missed on the first one?
I got them right here.
Oh,
We're gonna hopefully get to that.
Okay.
Oh,
Yes.
So
Yeah.
being that the
Could you
final
mind
exam is comprehensive,
Yes.
are we gonna have questions from previous units on next unit exams? So like on this is unit 2, right? So will we
This
have
is unit
questions
4,
regardless?
right?
Right, sorry, that's how my anatomy.
No, you're
class
broke.
broke.
Like, will you, like
we gonna
this
have
exam two
Is
had stuff in
everything
previous,
comprehensive?
no,
Okay.
no. So for this next exam, you'll just have this set of content. To understand this set of content, you need that previous knowledge, but you won't have questions that are specifically for that older stuff. And then when we get to the end, we'll have a final exam study guide. You should have time where I can bring all of your exams and we can go through them. You can sit and look through them all right before the final, so. That can be helpful too. I'll throw them away, so.
I kind of like want to cry right now. This is so.
Are you gonna ask us to draw the PTC cycle on the exam?
I may, yes.
Okay. Good to know.
It could just look like that though.
It could just look like this though, and you'd be
Okay.
fine.
If we were to draw it, would we have to explain it?
Have to label it.
You'd have to label it.
Okay.
You wouldn't have to give like a paragraph. I will typically say, you can draw it or explain it, right? Because some people would prefer the paragraph, yeah. Yeah, this unit is very material heavy, but the problem is that it's considered prerequisite content. So that's the issue, is that because the prereq for this course is a biology class, you should have, the preferred biology is 0100, 1111, or 1113. So you should have had this content and it should all be reviewed. So even though it's material heavy, it's supposed to be reviewed. The issue is that a whole bunch of people use anatomy or there's a loophole that allows you to use anatomy or physiology as a prereq to get in. And that means if you haven't taken a cell biology class, you don't really remember much of this one.
For me, it was my ACTs.
You used an ACT score.
I feel like you got that good on your.
Did you see some-
Mine was just in two thousand.
CCP, right?
I
Yeah.
think it was 2016.
Mine was 2009.
I told 11:13.
So that's
Not like riding a bike.
It's
not really like riding a bike, no.
But hey, it's all prereqs. so it's
And
fine.
it's honestly been the biggest battle we have fought, is that we want you need a cell biology class. But nursing requires this course.
Yeah.
Nursing is capped to a 65 credit hour limit for their program. So if we add 0100 or 1111 or something else, that technically increases their credit hour. And they can't do that from accreditation perspective. They have to drop another course. So nursing has actually told us we are not allowed to specify which bio class has to work, because it screws up their credit hours and it screws up their program and accreditation and everything else. So, and they dropped chemistry because they couldn't find a way to fit it in with the 65. And now they go, well, I know we don't have chemistry, but they had to make concessions once we switched over to semesters. Yeah.
Why don't we just make it? You have to have taken it.
They
Yeah.
can't. For some reason, they have more students who apply before they get to that point, or admitted before they get to that point, and they can't say it's a prereq for that either. They can't put it in. No, it doesn't. Trust me.
You know, new,
Matthew Selzler, yes.
I saw him the other day, he was like their last semester, and I think he's really nice.
He is very nice. I should pass him on the way over.
And also, so you're the head of your, like, area, right?
Yes,
Does that include him?
no.
Is he the,
He is the lead over general biology.
okay, because that's what I thought, because you'd say some of my adjuncts, but then I was like.
Yes. We used to be, him and I used to be together over everything, and it was just, it was too much. I hated it. So he now gets all those gen bio stuff and I just handle the micro. So, and the college credits.
You do college credit.
Yes,
What classes?
all of our biology in the high school, they're all mine. So I get to go to Big Walnut High School tomorrow. So,
What high schools do you work with?
so Big Walnut, Marysville Early College High School, Hamilton Township, Reynoldsburg, it's like a STEM academy over there. We've got 4 Columbus City schools.
I know Buckeye Valley did. It was my husband did college credit plus through.
Yeah. Yeah, we've got a few other ones where we do the biology. We're gonna do Dublin next academic year. They're coming in too.
Do you do Worthington?
We don't at the moment. We do, Bishop Watterson was one of ours, but not a Worthington.
Yeah.
So. they have to have they either have to have enough students that they can offer the whole class in the high school, or less students, but a qualified instructor who's there. And so that's kind of the trade-off. Because all the biology classes need this 2-hour or 3-hour lab, trying to get those to fit into a high school block is, that's normally the holdup, is that they go, we don't have an instructor who can take a 2-hour block off, so it's easier to send students here or to Dublin or somewhere else instead.
What my school would do. Oh, you wanna take it? Just go to campus.
Just go down there. We'll rearrange that. It's easier for a few students here or there than an entire instructor's schedule. So, yes. Marysville added it for school. So it starts 7 something in the morning. So I have to be out there in a couple weeks. I was
Do
like,
you like go to like one like district per week, or is it like?
it'll vary, but it's typically 1 per week. I'll just go observe them depending.
Are you gonna erase the ETC on the board?
No. Not till we're all done. Okay, and by the way, we've got all this conversation on, so that's good.
You know, the
All
people
right.
that aren't here are gonna be in the conversation.
People who aren't, they're gonna feel like they belong, so it's gonna be great. Right. All right, I'm just gonna let that go. How we feeling? Terrible. We're gonna briefly get through photosynthesis. I'm gonna post some little videos and other things to help, okay? In Blackboard, in the Unit 4 folder is a whole bunch of supplemental videos. There are some videos from your textbook. There are other videos that we made in-house a few years ago for our 1111 and our 1113 courses. So they are in eukaryotes, but they're more well done because we did them. So you will see Professor Selzer, he does one. I didn't do any for this particular unit, but they give you kind of just the components that you need. So they're really short little videos that walk you through it. Look at some of that supplemental information, and then we'll kind of keep reviewing it. Sound like a plan? We're good. We're gonna finish with photosynthesis, okay? I know, all right? And the problem is that some of these prokaryotes do photosynthesis too. So it's not, I know we only do respiration, but we're gonna cover it because we're gonna be working with microorganisms that do photosynthesis. Photosynthesis is unique because it is both catabolic, and it's harvesting the energy from the sun, and then it also is anabolic in that we make compounds in the end. One of the things you will notice from this equation, in photosynthesis, the general equation for this process is to start with carbon dioxide, water, and sunlight, but we produce glucose, okay? Glucose, water, and oxygen gas. Some of these processes will look similar to you, but they're almost kind of opposite in terms of what they produce. So you've got 2 sets of reactions that happen here. Some that are known as light dependent, which means they only happen in the presence of sunlight. And the other ones that are light independent, which means they can happen in the dark. They don't actually need the sunlight to occur. The light reactions make 2 components. They make ATP, and they're also going to generate reducing power. In this case, it's going to be reducing power in the form of NADPH. The independent reactions are going to take the products and they're going to make organic compounds. So we're going to take all the ATP, we're going to take all the NADPH, and then we're going to put them together to make sugar with it, okay? So here is a way of looking at the light dependent reactions, okay? These photosystems are molecules, okay? They have pigments, bless you. I'm sneezing, okay? They are pigments, and they are pigments that collect the light. So what happens here is that to start this process, a molecule of water is broken down. The water is broken down to become the electron donator into this process. So the electrons got to come from somewhere. In this case, we break down water to come into this process. When the light hits on the photosystem, it bounces all around and ejects that electron. The electron goes down a carrier, similar to what we talked about with the electron transport chain. Do you see how we have proton pump here? And it says proton gradient formed for ATP synthesis. This pathway here creates ATP. The electron goes and sits in another photosystem. More light pops in, the electron pops out, and it goes down another carrier system that makes NADPH, okay? So this is entirely dependent upon energy from the sun. And we're using that to still use a proton pump, but we're making ATP, and in this case, we're gonna make NADPH. You will see these photosystems. Photosystem 2 is the farthest one over here. Photosystem 1 is the next one. Photosystem 1 was actually identified first, and that's why it's called photosystem 1. They then realized, wait a minute, there's a part of the pathway that has to happen first because the water has to break down first to generate that electron. So this part was actually added in second, but you gotta make ATP and you gotta make NADPH. The more energy from the sun, this process just keeps happening and happening and happening and happening, okay? We doing okay? The products from those first reactions go into the next one. The next set are called light independent. They're also called the dark reactions. You will also see this described as the Kelvin cycle. Do you see how this is a cyclical pathway? Kind of looks like the TCA cycle, okay? However, do you see how carbon dioxide is coming into this process? 6 carbon dioxide coming in. We're gonna end up making a 6 carbon sugar that comes off of that. So every turn of this process is gonna add in a carbon. Adding in a carbon through this process is also gonna allow rearrangement of a phosphate molecule. So all the ATP that we generated before are all gonna be broken down here. All the NADPH are all gonna get broken down here in order to generate that sugar molecule. You're gonna break down more ATP to regenerate this process. So you need the products of the first one to do this. Yeah.
So if you need the products of the first one, is it added later in the cycle?
So
Like, so we need the products of the first one to do this.
yes,
Where do the products come in?
so the products from this one that are coming that way are the NADPH and the ATP. And then they're coming in, here is the ATP and here's the NADPH.
Okay.
And so these are light independent. They can happen in the presence of the light, but as you're generating one from that one, it flows right into this.
And then
It
they
just keeps
grab
going.
the carbon dioxide from something else.
Exactly.
Okay.
So yeah, the carbon dioxide, if it's an organism that is also capable of doing respiration, the carbon dioxide generated from PCA and the transition step can flow into here. So some organisms can do both. Other organisms like plants can do both. Plants can also bring in carbon dioxide from the atmosphere. Some microbial cells can bring it in. Some microbial cells can also do respiration and then take that other CO2 and then bring it this way. Yeah,
So where the carbon dioxide come from varies.
it's gonna vary. Yep.
Same with the ATP.
So that ATP will all come in from over here, but it will, how much comes is gonna be dependent upon how much sunlight's there. We doing okay? This one is a minor part of the process. If you got this other stuff down, this is not really your problem, okay? The whole goal of catabolism is to make energy and these intermediates to then go into anabolism. So you can take all of these precursors and other components and put them together to make other things. We've seen this part of the drawing before. However, what I'm showing you is that, hey, if you end up with too much of these products, they can be funneled to make other components. So amino acids, for example. Amino acids are put together to make proteins. If you've got a cell that needs to divide and it needs specific proteins, it can convert and funnel things away from this as well. So the cell doesn't go, I have to make ATP, I gotta go all the way through. It can kind of mix and match what it's doing with it. The terminology is half the battle. So if we're on a term or something like that, we don't know what it means, make a list, make some flashcards. So whether that's, yes, catabolism, anabolism, whether that's enzymes and they're made of protein, whether that's an electron carrier, make sure you've got the terms down, okay? Remember, when we say this metabolism, it's just a blanket term referring to all chemical reactions. Every chemical reaction inside the cell requires an enzyme in order to occur fast enough, okay? In lab on Thursday, we're gonna be doing some processes looking at enzymes. We're also gonna be doing some processes looking to see if they complete fermentation or respiration, and we'll look to see what their products are that they give off. You have to have a constant supply of ATP. So going all the way through or doing fermentation and making a small amount of ATP is required by the cell because you have to have that energy molecule. Every species is gonna vary in terms of how much ATP they make, the exact components they use, whether they're using aerobic, anaerobic respiration, fermentation, photosynthesis, what those electron carriers are, they all vary. I took an entire course in college. It was all about, you give an organism this compound, what do they do with it and how much ATP is made? Oh, it ran out of this. What happens to be made instead? It was microbial metabolism and something else. It was the worst class. It was a half class because we did something else the last time. It was a rough like 6-8 weeks. It was bad. And all you did was draw this out the entire time. And it was, you had to know for this organism what was made. Just understand it's going to vary and you're going to be good to go. Okay. My pain will not be your pain when it comes to that. Okay. All right. You can find a lot of videos. I'm not going to bring these up for you, but you've got them in the notes. You've got access to them. YouTube's got all kinds of good stuff. Good or bad, there's a lot of good stuff on these topics because they're covered in regular cell biology. However, for a eukaryote, it's going to happen in the mitochondria. The exact amount of ATP that's produced will be different. So if you can look at a video and go, okay, I know I'm just doing this for the process because the location will change and the amount of ATP will change, good for you. You can also go to YouTube and search glycolysis in a prokaryote, like be really specific with your terms. If you're watching a video and you're like, this doesn't make any sense, shut it up, shut it down and get a different one, okay? There are lots that can help you. This one for photosynthesis is a Z-scheme video. It was originally made at OSU. Jim Schressel is in the video. It's terrible, okay? But you watch, they got the football and the football acts like the electron and they pass it around and you, okay, it's a whole thing, right? I don't care what helps you learn the process and passing it around and where the electrons go. You just need to be able to visualize it and get it down, okay? So you can find all kinds of things. There are things in Blackboard as well. Find one of those that makes sense for you. Here is a video or a table from an earlier textbook we used to use. I like that one better for this chapter, okay? But it also breaks it down for what is the output and then where all those molecules are coming from. If you are more of a numbers person, I have to see the numbers. That's partly why I leave in the individual chemical reactions because some students are like, I gotta see the flow, right? If you are someone who prefers to look at the numbers and then add them all up, tables like this can be helpful as well, okay? You want to make sure you can kind of explain it. You can walk through it. If it says, here's the product that was made, where did it come from? You know, you are left with pyruvate. Where did that pyruvate come from? You want to be able to kind of oh, I know if it's pyruvate, I know it had to come off of glycolysis. I know if we're talking about acetyl-CoA, it had to come off of the transition step or pyruvate oxidation. Does that make sense? Okay. So work your way through it. That's the biggest suggestion I can make. Like this on the neck for the next exam, this is the biggest set of components that trip more people up. This is the one that's most time consuming. The diversity stuff that we're gonna get into next, you're either gonna know it or you don't know it. And some flashcards can really just make everything easier. It's a lot more memorization where this is a lot more process oriented. Yeah.
So we need to know for the ETC and prokaryotes. It's the same as eukaryote, just different location.
Different
The
location
eukaryote
is in the
and
mitochondria,
this one
right?
is across the membrane.
This is just across the membrane. Right. I'm really hoping we're still recording here. I think we are, but. Doing okay? This next set is literally taking your metabolism and applying it. So if you got the metabolism stuff down, this then becomes a lot easier, and it's then just about the terminology. Yeah. This is unit 5. Next week, we're gonna get through unit 6. Units 4, 5, and 6 are what's on the next exam. There are 2 chapters in unit 6, but it's all microbial control. Like, why do you heat things? And what does the word sterile mean? That is a lot easier. You know, the antibiotics we're also gonna talk about, it's, you either know what the antibiotic targets or you don't. It's more memorization. Metabolism is much more process driven. So unit 4 is the biggest one that you gotta get through for that next exam. Okay. All right. We've been through a lot now. Guys, believe it or not, we're gonna have next week. And then after next week, we're gonna take an exam and then we're gonna head into your immune system and how the immune system works. We've essentially covered, this is week 5, the main points of microbiology and all of the ways in which microorganisms grow, how they divide, what they need to survive. So everything from this point forward is really just finding ways to apply what we've discussed already, okay? We've talked about these prokaryotes. The last thing I said was, remember, all these organisms vary. The processes they use vary by species, right? There are an estimated 1 million species of prokaryotes, right? We can't possibly say this one works in the majority of them because that's not the way it works, right? Every species is going to be different. To date, roughly only 6,000 have been characterized. It's not a lot, but think about the things that we've talked about so far. Organisms can live way up in, you know, in the mountains where there's no oxygen, where we're not going, and it's super cold, and you think there's next to no nutrients there. Bacteria have found a way to survive. They are in the ocean, way down at the bottom where there is no sunlight, okay? And they are in some sort of, you know, thermal vent that is super hot, okay? So lots of organisms are present, but we've only characterized roughly 6,000 to date. We are finding new species all the time. That is new species identified in harder to reach locations. That is new species identified in easy to reach locations. Ohio State did a study. You're gonna love this. They brought in like over 1,000 students and they simply said, we want to swab your belly button. No joke. They identified like 3 new species that have never been characterized before. All they did was swab it and they grew it on some plates and then they sequenced them all to see what had been characterized and what had not been characterized and all the different, you know, species that they found.
There was
Okay.
a guy who had suffered with Japan and but he had never even left like his state, like, you know, he's from. That's-
Yeah, so it's like, you know, it's different practices, it's different, you know, all of these things are different, so you come up with different stuff. But we're constantly identifying new microorganisms. To try and arrange these organisms into groups, there are a lot of things that go into it. It's not just identification, but it's looking at what does the genome look like? So what is its chromosome? What do its genes tell you? It's where does it grow? It's what are those growth factors? All of that plays into this. Once we found a way to identify it, Then we say, okay, how are we gonna name it and what are we gonna use? Good or bad? Especially, years ago, everything went according to the person who found it, like. That individual wanted their name, remembered for all time, so they would put their name and the name of the organism kind of thing. Classification, once we've got somewhat of an idea of their characteristics, we can then start to arrange them and organize them into groups together. This process involves a lot of work. Yes, it is looking at it under the microscope, but it's also understanding its culture characteristics. What temperature does it like to be growing at? What is its optimal pH? Does it like oxygen, no oxygen, a little oxygen? How does that work? Biochemical testing are tests that are done to look at the organism's enzyme profile, or what can it break down, or is it doing aerobic respiration, anaerobic respiration? How does that work? Yes, you can do a nucleic acids analysis. That essentially means sequencing the genome. If it causes disease, what are the symptoms? What are the signs? Are they characteristic in most individuals? Do they change? What do we see? All of that goes into this process. You are doing part of this in the lab in the form of the unknown experiment. We're going to hand you a mixture of 2 organisms. You are going to you're going to grow it in culture. You're going to generate a pure culture first using the streak plate. You are then going to stain them and look at their gram stain characteristics. You are then going to perform biochemical tests to understand what can the organism do and not do. What is its energy source? What can it use as a carbon source? When you put all of that information together, you will be able to identify the organisms. We've done all of that up until now, except for those biochemical tests, and that's what we're going to start in the lab on Thursday, okay? So this process is common in lots and lots of different ways. I do want you to understand that trying to understand evolutionary relatedness in a prokaryote is super difficult. It's difficult, because of that binary fission process. 1 cell goes to 2 cells, 2 cells goes to 4. Remember, there's not a parent-child relationship there. It's not a familial relationship when you look at the genetics. Everything should be genetically identical. So because of that asexual reproduction, automatically right there, we're not getting some sort of differences that can be taken, like this is, you send your DNA to 23andMe and 5 years down the road, they've caught some, guy because you showed DNA, they matched the DNA profiles and they went through it. Not really possible here, okay? There are a few differences in cell morphology. You have seen this, right? Most of the things we look at in the lab are either a really awesome caucus-shaped circular cell or some form of bacillus. Okay, you can also be a spiral. That's it. We've characterized 6,000 of these organisms, and those are basically the 3 different shapes, okay? It's not like they stand out like a sore thumb to tell them apart. We can do sequencing, we can look at their genetics, and that does help typically more than anything else, is to look and see what genes they have in common and where those genes are different. But it's complicated. We looked at this the very first lecture, okay? When we are branching off, you've got the eukaryotes over here. I know we think about these archaea as being more extreme, but remember archaea actually branched off later. The bacteria became their own group earlier, and archaea actually have a decent amount in common with eukaryotes, okay? When we talk about these prokaryotes, there are characteristic categories that are important. Your textbook on this topic gives a lot of different organisms. You are responsible for the ones that are in the lecture notes only, okay? So if you're going through the textbook and you're like, we didn't talk about those, don't worry about them, okay? But we're gonna hit on several important groups, like lactic acid bacteria. Do you remember, like 20 minutes ago, we said something makes lactic acid as a result of fermentation, okay? We can actually group those. Lactic acid bacteria is a group of bacteria that pretty much exclusively does fermentation, and they give off lactic acid as a byproduct. We can group organisms that all form endospores. Remember, that's an optional structure that not all organisms can create. We're going to talk about anoxygenic phototrophs in a minute. Those are cool. They do photosynthesis, but do you remember how, a couple slides ago I said water is the source of electrons in photosynthesis? These guys go, screw that, we're gonna use something else as a source of electrons. We're doing photosynthesis, but I'm gonna use something else as my electron source, okay? Living organisms always have variability. There's not just a textbook way that these are all going through, okay? Typically, when we group them, they have some sort of similar phenotypic or physiological characteristic. They might not always be genetically related. So for example, those organisms that form endospores, those are not always all genetically related organisms. They can actually come in multiple different genuses. So their genes are not necessarily all similarly identical, okay? So this is like, taking us in here and saying, we're gonna group all of us now by who has a sibling and who doesn't have a sibling, or what color hair we have, or what color eyes we have. We can change those groupings depending on what our classifications are, okay? Same thing happens with bacteria. This information, remember, compounds on itself, okay? So you have to go back to your terms from before. Yes, we don't have specific stuff from those other units, but you got to remember the stuff from the other units in order to be able to apply it now. So I'm bringing up a table we looked at a longer time ago. Differences between carbon source and energy source. So your energy source you can get from the sun. You can also get from chemicals. Carbon source can either come in the form of organic carbon or inorganic carbon. Inorganic carbon means it's coming in the form of CO2. What you use for your carbon source and your energy source dictates what type of organism you are, okay? You have also seen this slide as well. Remember, we are here in terms of how long we've been around, comparatively speaking to most other organisms. Prokaryotes have been around roughly 3.8 billion years. They live almost everywhere. They have found ways to adapt to their environment and to create new ways of harvesting energy and using different compounds as carbon sources and energy sources. So we're going to break it apart. We're going to go through some specific groups. I'm going to give you examples of each type. Partly because these are organisms we use in the lab, but they're also organisms you have heard about in real life as well, okay? So anaerobic chemotrophs. Anaerobic, we got to be thinking no oxygen. So we're doing anaerobic respiration. If we can't do that, do we do fermentation instead? Okay. There are current anaerobic environments. This is one of those where everybody goes, well, isn't everything just aerobic? No. Okay. So deep down in a water column becomes anaerobic. Higher up in the mountains becomes anaerobic. Even in your body, you have anaerobic environments. So by the time you get far along in your GI tract, there is little to no oxygen that's left there. It is an anaerobic environment. Deep down in your gums, as the gum seals against the tooth, it actually closes off and becomes an anaerobic environment. Okay, so we've got lots of different organisms that can live in these different spots. So we can be a chemolithotroph, we can be a chemoorganotroph as well in those different locations. So things we might have heard of. So anaerobic chemolithotroph. We got to be thinking inorganic chemical now, right? This is where our terminology is coming in. These guys are only doing anaerobic respiration. So that process here, but instead of oxygen as the terminal electron acceptor, it's something else, okay? They are actually going to use an inorganic chemical as their energy source. They're not starting with glucose. They're going to break down something else to then function into this pathway at some part. As their terminal electron acceptor, they can actually use carbon dioxide. Some of them can use sulfur. Most organisms in this category typically are archaea. An example of this that you might have heard of are going to be methanogens. Methanogens actually bring in hydrogen gas as their electron source and carbon dioxide as their terminal electron acceptor. Okay, so different. But as a process, as part of this process, they release methane gas. They are found in all kinds of different locations, but a location you might have heard of is creating biofuel from mammal manure. There are places in Ohio that do this, okay? Specifically, cow farms, cow pastures, okay? They take all the manure and they actually push it into these giant pits. The manure is got the methane gas trapped in it. So they push the manure into this deep pit. They have tarps that go over the top. The methane gas all comes out of that manure and they collect it. And then that is then used as biofuel for all kinds of other things. Because it's releasing all this methane gas into this pit, those are actually anaerobic environments. All the oxygen is driven off of those pits. Occasionally when these come into the news is because some poor soul like falls into the pit and then they suffocate because they can't breathe. It's full of methane gas. It's not full of oxygen that they can actually breathe in. But if you've ever heard of, so there's a whole thing with cows, believe it or not, okay? Cows contributing to greenhouse gases and warming of the atmosphere. It's because of these bacteria literally living in their gut, okay? I don't know, it was a Dumb and Dumber. There was a movie, none of you have ever seen Dumb and Dumber, I guarantee it. There's a very old movie, okay? And they run up behind a cow and they light his rear end on fire, okay? The reason that works is because they're giving off methane gas. Cows have a farting issue if you like, okay? My kids always find that one super funny, okay? But it comes back to these organisms that are living in there. And they're giving off this gas that then does all kinds of other things in the atmosphere. Yeah.
New Zealand is trying to start taxing like cow farmers because of the methane.
See? Yeah.
There's so much.
There's just so much of it. And it's, you know, that then goes into the whole thing, like if we ate more plants and we ate less meat, that would also be better for our environment and all of these other things. Yeah.
I was watching this special about like climate change in general and how it relates to food, and it's shown that if you eat local meat, it's actually better for the environment, like carbon wise, than eating imported vegetables and fruits.
Right. Because it's coming from less and then the less travel is less carbon emissions from shipping it all around.
And they have some carbon negative cattle
They do.
farms
See.
too.
Well, and that's because they're also bringing in the methane. They're doing other things with it to try and neutralize some of those components. There are some studies where they're actually trying to change the environment of these cows so that they have different types of organisms in their gut and not these methanogens. They are testing different types of feed and how that equates to the methanogens and their production. There is an image, I should find it. It's a cow, but the cow looks like it's got like a front loader washing machine door on the side of the cow, like in the side of the cow. And they can open this door and they can go into those stomachs of the cow because they got like 4 stomachs. You can go in there and take out the breakdown products basically of everything they eat. And they look at that to see what is the composition of organisms that are in there. So how can we change that composition based on what they eat to try and make them produce less gas in the end? It's a whole thing, okay? But it's all coming back to these methanogens. So these tiny little organisms that we can't see are responsible for all kinds of things in our environment. An example of anaerobic chemoorganotrophs. Organotrophs, so we're thinking organic compounds, are going to be those from the genus Clostridium. We've talked about this one, Clostridium tetani, Clostridium botulinum, okay? Clostridium difficile, which makes C. diff. So all of those organisms from that genus Clostridium are all a gram-positive bacillus-shaped cell. They all are going to produce endospores, okay? When we get into talking about why tetanus is a problem, why you need a tetanus shot, it's all coming back to the fact that that organism makes endospores. C. diff, the reason C. diff patients get C. diff over and over and over again is because it makes endospores. You take the antibiotics can't target the endospores. So it kills off everything else. The antibiotic goes away, those endospores go back through a germination process and they colonize again.
This one and the one previous slide, the sunlight and the carbon sources.
So these are going to be anaerobic, but these are a chemoorganotroph versus the other one, which is a chemolithotroph. So
These one we have the previous one we have inorganic.
the previous one is inorganic carbon, and these are bringing in organic carbon. So they're bringing in their organic carbon, but these are also going to be organisms that produce endospores as well. Anaerobic, so they got to be found in some sort of anaerobic location. If you get tetanus, that organism is typically coming in because you step on something that came from the outside, came from somewhere. When you acquire that, you're acquiring the endospores because you're acquiring it in an aerobic environment. The only part of that organism that survives are going to be the endospores, okay? So the way that organism grows influences its disease-causing characteristics as well. The lactic acid bacteria are also a form of anaerobic chemoorganotroph. We're going to get into a lot of these. We work with, so the Streptococcus lactis, we use that one a lot in the lab. That is technically a lactic acid bacteria forming organism. Okay. As Pyogenes, we had this one out last week. We looked at it on a blood agar plate, created that hemolytic zone. Okay. It's classified as a lactic acid bacteria as well. So we can group them depending on what they do. All of these organisms almost exclusively participate in fermentation. And because they're doing fermentation, they're giving off lactic acid. All of these that we're showing you here all happen to be gram-positive. So remember what that means. It's got a cell wall that contains a thick layer of peptidoglycan, okay? This is really where the terminology is a big part of it. You know, if you look at it and you go, I don't remember what gram-positive means, you want to go back to those terms because they're going to keep coming up. Your differences between cellular structure and function are also going to keep coming back. You know, lactobacillus. Those are organisms that are typically, they're all found in normal flora, but they're all used in food production as well. So these organisms come in lots of different ways that we see them, that we use them, but we're grouping them in this case because of the fact that they all produce lactic acid. Yes, 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 there are microorganisms that do photosynthesis. I really, bring this up a lot because people tend to think, okay, photosynthesis, it has to be a plant. Doesn't have to be a plant. Remember, there are microorganisms that fall under this particular category. So in this case, there are bacteria that do photosynthesis, okay? So we have purple sulfur bacteria, we have green sulfur bacteria. These are ones that are anoxygenic. Anoxygenic means without oxygen. So they're completing photosynthesis for their energy process, but where they are getting those electrons is going to differ. In this case, they typically bring in hydrogen sulfide or some other organic molecule to do this. So they do not bring in water. They're bringing in some other compound to feed that process. These organisms typically are found in bogs and lakes, but they are truly bacteria. You see this image here. You would look at that and you'd go, oh, that's a bunch of algae, right? That's kind of what we've been taught to think. But in this case, it's actually bacteria. There are billions and billions of bacteria in this image that are all on top of one another, that are all floating at the top of that water column. You would think, okay, we are in a water column, so wouldn't it be really easy to just break down water as our source of electrons? You would think so, but they don't have that enzymatic ability. They've lost that part of the pathway, so they have to break down some other compound instead. Really remember, looks can be deceiving. Just because you see water here and you think, okay, they got to do photosynthesis, doesn't mean they're actually using that water as their source of electrons. You have other organisms that are oxygenic phototrophs. These organisms do typically live in a watery environment, but they can also be found in soils as well. So don't think, okay, something that uses photosynthesis has to live in an area that's going to get lots of sunlight. You can live in the soil and do this just as well, just as evenly, okay? These organisms tend to live in a sheath, or they tend to live under they encase them. So these guys are all individual cells that are all cased together in this coating that actually links them all together. It helps protect them from too much water diffusion process. These organisms are interestingly, typically these cyanobacteria are actually responsible for blooms of the organisms that release toxin. So if you have heard of lakes and streams, there are several around here in Ohio where they tell you, okay, don't swim in there anymore. Don't let your dog go in it anymore because there's a toxic bloom that's happening. It's actually cyanobacteria that are in the water that are giving off toxins as a byproduct of their metabolism. Those toxins accumulate in the water and they can be toxic to our functioning. A lot of times you will see those advertised as like an algae, toxic algae, but most of the time there's actually cyanobacteria that are responsible for that. If you see bacteria in the end, you want to think it truly is bacteria, not some sort of eukaryotic organism. This is the one that's the biggest category. So aerobic chemo organotrophs, we're using organic compounds for both our energy source and our carbon source. And in this case, we're telling you they are aerobic. So their preference is going to be aerobic respiration, okay? So we're kind of combining our terms to get that. This is the biggest category of bacteria that we work with. Humans also fall under this category. We as humans are aerobic chemo organotrophs, okay? So oxygen is its terminal electron acceptor. We're bringing in some form of organic compound as our energy source, just like what we talked about. So glucose is an organic compound. We can break down other compounds if we need to. These come in two different formats, obligate aerobes and facultative anaerobes. Again, terms you have seen before. If it is an obligate aerobe, it only uses aerobic respiration. Facultative anaerobes, they use aerobic respiration when oxygen is around. These organisms, however, typically switch to fermentation when there is no oxygen. That's the biggest, sometimes I think that's a big hurdle to get over, right? You think, okay, if I don't do aerobic respiration, I switch to anaerobic respiration. The problem is that these guys are so used to using oxygen as that terminal electron acceptor. All of their carrier molecules are set up for this one part of the process. So if oxygen is gone, they go, holy smokes, we don't know what to do. We're lost because the end part of this can't be completed. So they actually completely switch to fermentation instead. That actually becomes an easier process for them to achieve.
Can that be kind of fluid, like they can flip back?
They can flip back and forth based on what's around. That's kind of the cool part. Gives them a lot of variability and a lot of ability to grow under all kinds of different conditions, like the E. coli that we looked at last week. Remember, it was really growing like crazy on some of those plates. And I was like, this one has no issues growing. It's a facultative anaerobe. So it can pretty much survive on next to anything. And even when oxygen is limited or glucose is limited, it shifts over to something else instead. So examples, Micrococcus. You guys saw this in class last week. We're going to have it out again this week. It was the Micrococcus luteus. It was turning yellow on that plate. Remember, it was super teeny tiny colonies. These guys, they're gram-positive cocci, but it's truly an obligate aerobe, okay? It makes this really nice and orange kind of pigment to it. The pigment is actually temperature dependent, but it's only going to grow in an aerobic environment. If we were to take that Micrococcus luteus and toss it in an anaerobic jar, it's going to die, okay? Yes. You have Mycobacterium. We've talked about this one, Mycobacterium tuberculosis. Remember how we looked at this one because it was acid fast? We had this one out when we did the acid fast stain. It's got those waxes and lipids in its cell wall. It's gram positive, but it's truly an obligate aerobe. If you think about someone who's got TB, a tuberculosis infection, where is that located in your body? In your lungs, right? Your lungs are full of oxygen. So it kind of makes sense that this one is going to grow in a place that's got the most oxygen because it's an obligate aerobe. Pseudomonas is another one. We're going to get into this one a lot more later on in the course. This one happens to be gram negative. It's a bacillus shape, but it's common in soil and water. It is a really common opportunistic pathogen. We did have this one out. This one is common in burn victims. It typically colonizes when your skin, you've got a large portion of your skin that's burned, it can colonize the skin surface. This one is also going to be common in ventilators, believe it or not. So it's found in hospitals a lot of times where the ventilators aren't fully cleaned. And it can actually survive in soap. It can survive some disinfectants. So it becomes a real big problem if you've got a patient who's on a ventilator and got other issues. Yeah.
So someone's like intubated, that one needs to.
Yes, that's exactly what happens when they're intubated and they end up with pneumonia typically. It's typically almost always Pseudomonas. Facultative organisms. Facultative organisms can live in all kinds of different locations because they can do aerobic respiration, they can switch and survive without it as well. Most of the organisms that live in your gut are facultative. When you think about the gut, parts of the gut are aerobic and then parts of the gut are more anaerobic. These organisms can also survive outside of the gut. So that's why being a facultative organism really kind of is best in that particular location. So whether you're talking about Klebsiella, Proteus, E. coli, all of these are normal flora organisms. One of the things that you might notice as we talk through these things in this particular set of lecture notes is that a lot of the stuff we work with in class, they're all normal flora organisms. These are not complicated organisms or things that are hard to find. We could isolate them for most of us if we found the right location, okay? You know, so they're there. Remember, they do good things there. You want normal flora in your gut. There are some of them that can be medically significant. Salmonella species is one of those that is common in the gut that can also lead to infection and disease. E. coli is one. E. coli is great when it's in your gut, but when it gets at the wrong place at the wrong time, then it's creating E. coli-based food poisoning, and nobody wants to have lots of vomiting and diarrhea, okay? It's just about that organism getting in the wrong place at the wrong time, okay? If you've ever heard of this term coliforms, coliforms represent intestinal bacteria that specifically ferment lactose. Last week in class, we had the McConkey's agar plates, remember? And one of those organisms, the E. coli, turned that magenta color on that McConkey's agar plate. And we said that means it's a gram-negative enteric, and turning that magenta color also means it ferments lactose. So coliforms are actually used by regulatory agencies to measure fecal contamination or pollution of items. So your water, for example, your water is all tested regularly for coliforms and presence of these bacteria. If you go to a public pool and they come out and they randomly check all the water, right? They're required to do that by law several times a day. They're looking for the presence of those organisms in the water. If they find them in the water, they need more chemicals, right? Or, you know, some little kid pooped in the water and now we got to wait for everything to, right? Everybody's got to get out for 30 minutes while the chemicals work. It's all coming back to this, okay? Small amount of these are actually acceptable in your water. Okay. But for the most part, they're kept in very, very low numbers because we don't want the water supply to be contaminated with something like E. coli. You're drinking it, you're brushing your teeth with it, you're bathing in it, right? It's just asking for you to get sick. So these things are commonly sampled. You can buy a kit to sample it at home. You go to home, you can get them free too. Go to Home Depot. On your way out, there are like free water testing kits. They're looking for this stuff in your water. So the chlorine will kill off those organisms. So it'll just kill them out of there and then it'll test negative.
So we have E. coli in our guts.
Yep.
So why do, like, I get if it's gonna make you sick, if you get it in a cut or you like to breathe it in or something.
Right.
Why when you eat it, does that make you sick?
So it's typically gonna die in other locations, but when it gets into your stomach and then it goes into the other parts of the intestine, it can actually release a toxin that then makes you sick. So in one location, it's not causing that problem, but in another location, it can cause you to get sick.
So where is it?
So it's normally in your gut.
What's your gut?
So your large intestine.
Okay.
So it's normally sitting in there. But if you've eaten something that's fecally contaminated, then it's coming in higher up. And some of that can survive the stomach acid and colonize the small intestine, and then you end up with other issues.
I was thinking of the gut as like the whole stomach.
Yeah, oh, gotcha.
So that's why I
So
was
different
confused.
parts of it, you will end up with different levels of where is it at and what's happening and all of
That
that
makes
stuff. Okay. Wrong place, wrong time, this kind of a thing. We're having fun yet? They can, you get someone who wants to sell you a water filtration system. They will sample your water and they will tell you all of the different things. You got to ingest a fair amount of E. coli in order to get sick. I'm just gonna say that. Same thing with salmonella, like a lot of salmonella before you're gonna get sick. Not that I'm encouraging that, I'm just saying. It's not like one E. coli is gonna cause you to have lots of vomiting, diarrhea, okay? Keep in mind what lives in the soil is gonna be different than what lives in a watery environment. Organisms that live in terrestrial environments have to be able to survive rain and drought and freezing and all of these other components. Is there a question? Okay. Is what? Okay, ask away.
Every time like you get. that's
Yeah.
usually what it
It's typically E. coli or salmonella is almost always what it is.
is. Somewhere here. Yeah, it's every-
Really?
Oh, yeah, I get.
Yeah,
I
I
guess
think
it's funny.
I think I've only eaten there once and I probably never will again. It's normally, most commonly it's E. coli or it's gonna be salmonella. Subway is probably E. coli. And it could be the lettuce or somewhere like that. Just when you think of, nobody here works in Subway, do they? It's washing your hands or not washing your hands, and then it's the raw vegetables that can have it on there. Exercise 16, the very last lab, I'm gonna bring in some food and you're gonna get to see what's on the food. And we specifically do a coliform plate and it will only grow those organisms that are on there. So I like to bring in spinach. Spinach is my favorite. But we typically do like chicken skin and ground beef and you get to see that it's all over the place. And then you don't want to eat raw vegetable skin. I'm good? Yeah.
Wait, you don't wanna eat the skins of raw vegetables?
Well, you don't eat the skin of chicken raw, but raw vegetables, there is a potential, depending on how it's grown, that it's fecally contaminated.
But if you wash it, it's cool.
Depends on how contaminated it is. Yes. Your risk is low, but it still is existing. And next week, we're gonna talk about microbial control and how to clean stuff, and then we'll be okay. What were we gonna say? Okay.
Oh, yes.
No. No. And I was like, then we have the conversation. Okay. What's that? I wouldn't put that on your chicken, but you could use that to clean after.
Oh, yes, I've seen people like on chitlins with. She's crazy. And then
Then
you turn white, I was like, oh my gosh.
Yeah, you don't need the chlorine and everything else, just.
I don't think I'll eat chitlins at dinner. Sorry.
That's the other point.
Yeah.
All right, let's get back to this so we can finish this and I'll give you your exams. We can look at that before I go, okay? Organisms that live in terrestrial environments have to be able to survive all of those changing climates. So lots of them will do other things, like they will produce endospores. Look, this looks like a nice thing of broccoli here, okay? They can make these what are called fruiting bodies, right? And it actually it produces a kind of a sheath, a structure that actually protects it in times when conditions are not favorable. What I really want you to remember is that these organisms have been here for forever. They're not going anywhere just because a little snow comes in or some ice. They have found ways to survive, and it is what it is. If they couldn't survive in those locations, they would not live there to start with. When it comes to aquatic environments, same thing. When you live in a watery environment, you have to be thinking about movement of water in and out of that cell, okay? So lots of organisms that live in aquatic environments actually bundle underneath a sheath. So you can see these individual bacterial cells. They can actually secrete a sheath that makes a full-on coating around all of them that protects them against lysis, okay? So organisms that live in watery environments as well also have to have structures that allow them to really survive. Yeah.
The sheet, is it like across the whole thing and just like section?
It typically goes across the entire string. Sometimes you'll get it where one cell didn't fully make enough of it and it looks like there's a little bit of a gap in between, but typically it goes all the way down a giant chain of them. Organisms that live in aquatic and organisms can do all kinds of awesome things. There are some bacteria that actually naturally bioluminesce, okay, believe it or not. So these are naturally glowing organisms. These are not like the cats with the glowing fur that you've seen on the internet or something like that, okay? So they naturally bioluminesce. This one is cool. It's called a flashlight fish. And this flashlight fish has an organ underneath its eye. The organ is actually colonized by these bioluminescent bacteria. This guy lives deep down where there is really no sunlight. And those bioluminescent bacteria give it enough of a light or glow that it can actually see where it's going. So it is a completely mutualistic relationship between these two organisms. And it's just the natural way that that one organism, those bacteria grow.
Are
Yeah.
they born with it? Or
Yeah.
do they have to get it?
So it gets colonized over time, but it's naturally going to be in the water and near them. So it will build up. Our bodies present all kinds of unique habitats for microorganisms. There are actually more bacteria on and in your body than you have human cells on and in your body, okay? You have a wide variety of habitats. Your skin is dry, it's more salty, the temperature is gonna be different. So, going back to that, let's sample your belly button and see what's there, right? It typically has a higher moisture content. It is typically warmer than something that's on your skin. So sampling different locations, even from the skin, you're gonna find a different composition of microorganisms. Some places in your body are free of microorganisms, like, your brain, your blood. You shouldn't have things there, but you want to think of your gastrointestinal tract as almost like a giant tube. So that giant tube in the center presents, that's a great place for microorganisms to live, okay? Your gastrointestinal tract, your digestive system, all of those present areas where we can get microorganisms to come in. Remember that organisms, because of those differences in growth, will live in different spots, okay? So just keep that in mind. The conditions are not the same throughout your body, so what lives where will vary. This can be parasites that live inside of certain cells. This can be organisms that inhabit membranes or in other places. Don't worry about memorizing all of the different organisms here. I want you to understand that our body is just like a breeding ground for bacteria. Okay, same thing with all kinds of other animals. Yeah. Your brain should be microorganism free. Yes.
Is that why when you get the things like mad cow disease, it's so bad?
That one is bad because there's nothing inhibiting it, and your immune system can't really get ahead of it. But anytime you get an infection on the brain, that's going to create swelling on the brain and a whole bunch of other problems and stuff like that too. So that one is one of those that's normally sterile. Don't memorize tables, okay? I want you to understand that there are tons and tons and tons of bacteria, okay? Some of these have a medical significance in just that they're normal flora. Others can actually get in the wrong place at the wrong time and make you sick. It really just depends. Don't think about these as like, I gotta freak out. Every one of these is gonna give me some massive infection and I'm gonna be in trouble, right? I guarantee you, all of you have had some level of infection before. Everybody has had some amount of food poisoning. You ate something that had organisms on it that caused you to get sick. The goal is to really understand that there's a lot of variability that comes with this. These are all gram-negative rods. Some of them cause more disease and others don't. They're just normal flora. Or some of those are organisms that can get in the wrong place in the wrong time. Klebsiella pneumoniae can be normal flora in the intestinal tract. Not in everybody, but it's possible. But then it gets into your lungs and it causes pneumonia. So it's got to be one place it's fine and one place it's not. Same thing with things like those that are gram positives. It's all about what they are. These are ones that are medically significant. There are thousands and thousands of other ones that aren't going to be capable of causing disease in any way, shape, or form. Okay. Remember, those organisms that live in extreme environments have to be able to survive those. Most of those are going to be all of in that archaea category. The biggest one of those are going to be those that live in high salt or high temperature. Anytime you see halo in the front, halo is referring to salt. Okay, we are gonna work with, couple weeks from now, we're gonna do an exercise where we put them in different salt concentrations and then see what grows and what doesn't grow, okay? You know, those methanogens are considered archaea. You've got extreme temperature ones that are found there as well. Just remember archaea live in extreme environments and you're gonna be good to go, okay? I can't stress this enough. Microorganisms are diverse. You're gonna get to know the common ones we work with in class, but don't freak out about something you've never heard of before, right? There are times where I read about something and go, that one is not one I know or remember, so let me Google it and see what its characteristics are and what its classification is. Microorganisms are diverse. Remember, it's coming back to those growth conditions. Where can it survive? What does it need? What is its carbon source? What's its energy source? What has to be done? Terminology is the battle here. You have to learn the terms. If it says aerobic chemoorganotroph, do you know what that means? Or an anoxygenic phototroph, do you know what that means? Don't get stumped on an exam question because you don't know the terminology. So my suggestion is to really make some flashcards and go through and really get those terms down. You can make flashcards with examples of organisms in each group. You want to just try to familiarize your stuff, you know, endospore, acid fast, gram positive versus gram negative. Those are all terms we've discussed before that all come back to cell structure and how it functions. If you don't remember those, you're gonna have a hard time then applying it later. Does that make sense? How do you see questions on this type of content? You can see questions asking you, you know, here's what's growing. Do you think it's a, you know, aerobic or anaerobic organism? Or what do you think the most common classification would be? You could see an example where it says, okay, we understand what's going on here. What is the characteristic that most supports that? Okay. So in this one, soil often goes through periods of extreme dryness and wetness. What characteristics of Clostridium make them well suited for those conditions? What does that one organism do that would make it survive? Endospores, exactly. So thinking through those characteristics, what do we do? What do we have? What allows it to survive? Grand Lake in Ohio. It's an algal bloom of bacteria and algae. You know, then you get into the broader prospect of this. You know, what conditions allow that to happen? And how do we change that? What do we do? Those are organisms that do photosynthesis. Can we control the sun? No. So what do we have to control? If we can't control the energy source, we got to control the carbon source and the other components that are there. Typically, these guys overgrow when things have been contaminated. When you have large amounts of fertilizer put on farms and you know, everybody's grass and stuff like that, and it ends up in the stream, you get all of this runoff water that then ends up contaminating a lake like this. It changes the population of some of these really basic elements. So nitrogen levels move. You can get them, you know, because nitrogen moves, ammonia moves, and you get all of these breakdown products. Sulfur gets in there as well. Phosphorus gets in there. When those nutrient compositions change, that influences what grows there and then what they're doing and what products they're producing. So yeah, we know we can't do anything to change the sun, but we know we can control what we're polluting it with. And we can try and clean up the water to try and bring down some of these other things that are there. What do we think? Stop recording. Yes. If there's any way, my gosh, this actually works, I'm gonna be really happy.